and welcome to our webinar series, Molecular Tools in Genetic Diagnostics, the application of cutting-edge biomarkers. In today's webinar, we have with us Brittany Pryor, MS, serves as a medical sales liaison, assisting the US sales team. Brittany brings nearly 10 years of experience as a clinical genetic counselor. During the webinar, she will discuss the following topics: insight from a clinical case, molecular diagnosis, and personalized treatments. If you have any questions, please enter them into the Q&A chat, and we will refer back to them at the end of the presentation. It promises to be a very interesting webinar, so let us begin. Brittany, the floor is yours.
Thank you and welcome everyone. Thanks for joining us. So as stated, we're going to review our clinical case, the molecular diagnosis, and then diagnostics management. So just some background about our particular case. We received a sample on a 10-month-old male, negative family history for any similar findings, and no consanguinity reported in the family. Perinatal history was fairly negative, child born at 35 weeks by elective C-section due to low fluids and a breech presentation, and had jaundice that developed at 4 days of age, treated with light therapy, but was otherwise healthy. Until around the age of 6 months, when the parents started to note the large abdomen and some motor delays. And by the age of 10 months, the patient then had low muscle tone, large pigmented blue spots on the skin, and some slight facial differences. The patient also had some additional studies which made it suspicious. There was enlarged spleen and liver seen on an abdominal ultrasound, explaining the distended abdomen. The biochemical testing showed elevated liver transaminases, cholesterol, and triglycerides, and there were some macrophages detected on bone marrow aspiration and trypsin biopsy. So this made a clinical suspicion of Niemann-Pick disease, which is why they then test the sample. So just a little bit of background about Niemann-Pick disease. There are 3 types of Niemann-Pick, A, B, and C, and they fall into a group of lysosomal disorders characterized by lipid accumulation. And clinically, they usually present with large spleen and liver. There's a component for lung disease, CNS involvement, and foam cell infiltration into various tissues. And they occur in usually 1 to 50,000 to 150,000 births. All 3 are recessively inherited and span over a wide variety of ethnic backgrounds. However, there are some ethnic backgrounds that it may be a little more common. So for type A, individuals of Ashkenazi Jewish inheritance may have a higher incidence, and in type B, individuals of North African, Saudi Arabian, and Turkish descent may also have a slightly higher incidence. And the clinical diagnosis is challenging due to a variety of symptoms that present at different ages. So a clinical diagnosis cannot be made alone. We need help from biochemical and molecular technology. And these 3 types are genetically heterogeneous and could be caused by mutations in SMPD1, NPC1, or NPC2. And just some further background about the different types. Types A and B are caused by specifically mutations in the SMPD1 gene. Type A is the most distinctive, meaning that onset happens very early on in the neonatal period. Usually the large spleen and liver is present by 3 months of age. And so any type that is not clearly defined as type A then falls into type B category. And so type B does represent a very broad spectrum of somatic and neurologic features, just of varying severity. And then type C is caused by mutations in 2 different genes, NPC1 and NPC2. The majority of individuals actually fall within NPC1. So type 1 is associated with gene 1 and type 2, gene 2. And over 90% of affected individuals have a mutation in NPC1. The classical clinical presentation for type C involves a mid to late childhood presentation with onset of ataxia, vertical supranuclear gaze palsy, and dementia. And just some further delineation in regards to how the types are different. So type A and B are classified under the sphingolipidoses. They are a defect in acid sphingomyelinase, and it's caused by accumulation of sphingomyelin and other lipids. Whereas type C presents differently. They fall under the integral membrane protein disorders, and it results in intracellular cholesterol accumulation, GM2 and 3 gangliosides, lactylcerylceramide, and glucocerylceramide accumulation as well. And here, this diagram really just depicts some of the difference based on onset and severity by type of Niemann-Pick disease. So type A, as mentioned previously, is the most severe. Death occurs in early childhood, typically by the age of 3. However, the mean age of death is 27 months. Type B, again, since most of type B is anything in the SMPD1 that is not clearly type A, has a lot of variance, and these individuals can survive into adulthood. Whereas type C can have a variety of presenting ages. The adult type is known to have a slower progression, whereas the neonatal type C does also result in early death and is more severe. And then by type, just kind of reiterating again that types A and B are caused by mutations in the SMPD1 gene. We can also look at enzyme specifically and a biomarker lyso-SM509. As well, type C is due to changes in NPC1 and 2. We can also get some information from looking at the staining of cultured skin cells and fibroblasts that could help us, as well as the same biomarker. Now, Centogene does have a ratio that exists to help us separate types A and B from type C of Niemann-Pick. So we can actually look at the ratio of the lyso-SM59 to lyso-SM465 to differentiate between those two types. However, types A and B, to differentiate between those is just based off of clinical presentation. And here, this slide is just a little bit of background from our internal database at Centogene called CentoMD. We queried the database for patients receiving a diagnosis of Niemann-Pick disease between December 2015 through August of 2019. So clearly here, the majority of the patients we saw fall into the Niemann-Pick type C category, and then specifically type 1. And we do have the largest cohort described to date, which does provide a valuable resource for discovering novel correlations and just, more information about the disease. Our median age of diagnosis was 3.6 years, and the range of diagnosis was anywhere from 0 to 76 years of age. And a review of the HPO terms for these patients. In a large majority, so over 76% of patients presented with enlarged liver and spleen. Almost 28% had a form of intellectual disability, and jaundice was present in 19% of those patients. So moving on to discuss the molecular findings of our particular case that was presented. So we did genetic analysis of the 3 genes, since at this time just Niemann-Pick was suspected and not necessarily one particular type. And we found 2 variants. On the right here, the diagram depicts that at position 725, we saw G to A substitution, and then the following T to G substitution. Parental samples were not available in this case, so that did limit us in being able to determine at that time whether these were in cis or trans. So just some more information about these two variants. They were both absent from public databases, as well as not seen in our internal database, CentoMD, and the predictor models were inconsistent. So at this point, we could really only classify these variants as variants of uncertain significance. So by ACMG reporting guidelines, we were still in the variant of unsignificance category because we could only identify these particular points. And so we needed something that could potentially help us get to a diagnosis or to confirm as the clinical condition was high. So we were able to say, could we potentially add some functional studies that could then help us change the classification of these variants? So most importantly, the sample that was submitted was sent to us on a Cento card, which for those of you who aren't familiar is our dried blood spot card. So the functional studies were not requested by the physician, but we were allowed to perform them as needed based off of the consent that was provided by the family and the fact that we had the Cento card sample. So we performed the additional biochemical analysis, specifically the enzyme activity for acid sphingomyelinase, and we saw that it was decreased compared to normal individuals. And then we were also able to look at the biomarker associated with Niemann-Pick and saw that it was elevated much higher than healthy controls as well as carriers, as seen diagram on the right. So by combining the studies here, we were able to then reclassify those variants of uncertain significance to likely pathogenic. And we were able to make the corresponding diagnosis of Niemann-Pick type A based on the clinical presentation as well as all of this data. And now just to review some more information about treatment and Niemann-Pick. So specifically, again, just to review some of the differences between types A and B. Type A is definitely more severe in comparison to type B. Type A, as mentioned, there's going to be normal development within the first few months, but then development does not exceed a 12-month-old level, followed by neurologic deterioration. And the findings in large liver are typically seen by 3 months of age and do become very, very large. The eye findings, the cherry red spot, are seen in all patients, maybe not necessarily right away, but they are eventually present in type A, whereas type B has a much more variable presentation as compared to type A. And typically, patients can live to see adulthood in type B, whereas type A, this is definitely a fatal disorder. And there are some genotype-phenotype correlations which can help clinicians. So on the left, these 3 specific mutations have been identified as being seen in type A, so involved with infantile form, whereas the 2 in the middle can be seen in types A and B, so it may depend on a combination of their specific mutations and the clinical presentation as to and how they are ultimately classified. And then the 3 on the right are considered neuroprotective, so they are definitely associated with type B, and these 3 mutations specifically have no neurodegeneration that has been associated to this point. There are a few other just based on ethnic background, genotype-phenotype correlations that can be helpful for clinicians. Specifically, an arginine 10 deletion, those patients have a much milder course. They have normal height and weight, a milder degree of the liver enlargement, and normal serum levels, whereas a couple of Saudi Arabian mutations seen in the early onset form. And of course, the treatment is going to differ a little bit based on the type that the patient presents with. So in type A, because it is a disorder, you're just kind of offering routine but palliative care. You're not going to be too aggressive with the treatment because the clinical course is going to be the same. There have been a few trials of liver transplants in children with type A that have been not successful at all. And then type B, you're going to have a routine follow-up of these evaluations at least once a year, and some of that may be dependent upon the particular presenting symptoms of the individual as to if they follow up any sooner than that. There is an enzyme replacement therapy, olipudase alpha, that is available. However, it has not yet been approved by the FDA. And for individuals with type B, there is a bleeding component that can be life-threatening and require blood transfusion if it gets to that point. However, total splenectomy is not recommended, as that can exacerbate the pulmonary disease for those particular patients. And just a little bit of review, since 3 types of Niemann-Pick are recessively inherited. So for any affected individual, they inherited both of those non-working copies from their parents. Since their parents have one working copy and one non-working copy, they actually don't present with any symptoms of the condition because that working copy is sufficient in their cells to make enough enzyme. Carrier testing is available for families once both mutations have been identified in a family, and could then be offered for family planning or future pregnancies, and as well for any family history indicative, you could do prenatal diagnosis using enzyme activity tests as well. So in summary, we had a patient with a clinically suspicious phenotype for Niemann-Pick disease. And considering just the genetic findings, we would not have been able to definitively say that was that patient's diagnosis. However, when we were able to add in the additional studies, that we could change the classification of the variants. And so those biochemical analyses were critical for the patient. And ultimately, it's important to remember that by checking the box to allow Centogene to store and use data, along with the fact that this patient has utilized a Cento card, is ultimately what allowed us to help add on these additional biochemical testing that was able to change the classification for the patient. So remember, it's important to provide consent for research, as well as samples may be used to help us further our biochemical research and ultimately, eventually, turn into new discoveries for other disorders. So thank you for your time, and I'll begin to take any questions.
Thank you, Brittany, for sharing your knowledge with us. Let us now move to the questions and answers part of the webinar. We have already some from our participants. The first one is, if I suspect acid sphingomyelinase deficiency in my patient, what is the recommended progression of testing?
So the consensus statement is that, so unlike in this case, that biochemical testing should be considered first, because as we saw with our particular example, that there are frequently variants of certain significance identified, and it takes ultimately a combination of biochemical and genetic to arrive at the diagnosis for the patient. So, enzyme first, followed by the genetic screening.
Thank you. Another one, they ask, how quickly will I be able to receive the SMPD1 DNA results of my patient?
So that does depend on which test has been ordered. So for sequencing, that's about 15 business days or 3 weeks. Whereas if it's ordered on an NGS next generation platform, that's going to be about 25 business days. So we had the question about whether ACMG guidelines have been inoculated in Varsone clinical software for variant detection. And I unfortunately am not well versed on that particular subject, so I can definitely follow up with you in regards to the answer for that. The question was, if sent a card was not originally sent, but only buccal for a whole genome or a whole exome, could we send in a sample at a later time if it would help with classification of a variant of uncertain significance in metabolic conditions? So I definitely think that that would be on a case-by-case basis. And we could discuss that based on the particular findings of that case, but it's not something that we could automatically always ensure that would be offered, and it might potentially come at a cost to the patient.
Thank you, Brittany, for sharing your insights, and thanks to all the participants for joining our webinar. We hope you were able to gain useful information on this very important topic. And right now we are going to launch a poll to receive your feedback on the webinar. So please take a few minutes to fill it out. And thank you one more time. Have a great day and stay safe.